So folks, I'm back. Here it is.
Okay.
Each of you, what a group. I wouldn't be here if you weren't here. And I love being here. And I think that you do too, because we all find so much companionship and the proper information that we're all looking for. So one more time, I just want to wish you the best of health. And I hope you find many answers here to make that come true. And with that, Lisa, it's all yours.
Welcome, everyone. Thank you so much for joining us tonight. I'm Lisa Deegan, and I'm part of the Everything ALS team. I lost my younger brother, John, to ALS about 4 years ago. So we love to provide this community access to the top experts, and we also like to provide the experts access to our community so your questions can get answered. And after the presentations, we open up where the recording is off, and we allow a safe place for people to talk to one another who have all kind of been in the same journey, walking in the same shoes. So thanks again for joining us tonight. And for anyone joining for the first time, Everything ALS, we are focused on advocacy and research. The research that we conduct is focused on prognosis and diagnosis utilizing digital biomarker assessments for the use in clinical trials. So the goal is to measure progression and if a drug or therapy is actually effective and working. So we need to find biomarkers that can be used in clinical trials. This will help us with earlier diagnosis and earlier treatments, which will be better patient outcomes. So if you have any questions or you'd like more information, please visit everythingals.org/research. So now for tonight's presenter, we have Dr. Matthew Miller. He is an associate professor, an associate chair, and an assistant dean in the Department of Biochemistry and Biomedical Sciences at McMaster University. He holds a Canada Research Chair in Viral Pandemics and is currently Secretary Treasurer of the Canadian Society of Virology. Dr. Miller's research has been focused on issues related to influenza virus pandemic, being prepared, especially vaccinations and the impact of viral infections, of the onset and progressions of neurodegenerative diseases, especially ALS. He's been awarded the CIHR New Investigator Award and several other awards, and he has been awarded from the government of Ontario, which is really big. So since 2020, Dr. Miller has been heavily engaged in COVID-19 research, including the development of next generation COVID-19 vaccines, therapeutics, and diagnostics. So please join me in welcoming Dr. Matthew Miller.
Thank you very much for that introduction. And it's a real pleasure to be here speaking to all of you tonight. So thank you so much for the invitation. I really, really appreciate it. I'll start sharing my screen now. And so just by way of background, obviously, I sort of come to this field with maybe a bit of an unusual trajectory, being someone who's real expertise is in the area of infectious disease and immunology. But I think what drew me into ALS was a recognition, I think, of the profound need to understand the disease from a much more multidisciplinary lens, and to ensure that people from different fields were working together to answer some of the questions that have eluded us for so long, and that I think have really hampered the development of the types of super effective therapeutic interventions that we would hope to improve the lives of people who are living with ALS. So the story I'm going to tell you about today is work my lab has been doing maybe over the last decade or so to try and better understand the influence of infection and inflammation in the context of ALS. And the presentation has a lot of data, but I'm going to make it really accessible. I'm going to try and sort of stay away from a lot of jargon and just really focus in on the key points and the implications of those points. And then I'm really looking forward to discussing this work with you further at the conclusion of the presentation. So, of course, the problem of ALS will be evident to everyone in the audience today. I think what a lot of people who haven't experienced ALS either personally or indirectly via loved ones is that it's the most common motor neuron disease. Often people talk about prevalence of ALS being, in the order of 1 in 100,000, but I don't like to talk about ALS prevalence because I think it masks sort of the true impact of ALS, because the lifetime risk, which is roughly 1 in 350 to 1 in 400, is actually, in my view, a much more important number than the prevalence. Obviously, one of the major problems in the context of ALS is that we still haven't come to a point where we have the types of effective therapeutics that we would hope for to alleviate disease. And obviously, there's a very strong genetic component. 10% of ALS cases are familial, 90% are sporadic, but these numbers really just have to do with whether or not ALS is in your family. It doesn't really speak in any meaningful way to the underlying biology of disease, nor the underlying sort of genetics. In speaking about genetics, obviously our understanding of genetics in the context of ALS has really exponentially grown in the age of whole genome sequencing. SOD1, which is going to be really the primary focus of what I talked to you about today, was the first gene validated to be associated with ALS back in the 90s. But since that time, there have been many, many more genes strongly associated with ALS. And what you can see here is that most of these genes have been associated with both familial and sporadic forms of ALS. And I think this is why, as sort of a scientific and clinical community, the utility of these terms to define ALS is sort of modest at best. It doesn't tell us a lot about the disease when we classify it as familial versus sporadic, because the underlying factors associated are often shared. What I think is one of the most sort of pressing mysteries of ALS is that it's so heterogeneous, both in terms of the way the disease itself presents, but also in terms of age of onset. So what you can see here on the bottom of this chart are a bunch of genes that are strongly associated with ALS. And the average age of ALS onset for individuals who have mutations in these genes. And you can see that this line jumps up and down all over the place. So there's a lot of heterogeneity in the timing of when disease presents, dependent upon the specific mutation that an individual has that's associated with disease. Of course, the vast majority of individuals who have ALS have an unknown genetic basis of disease, and that's probably a function of the fact that in many cases ALS may be polygenic, meaning there are more than one gene involved, or that the gene involvement is so rare that it's been hard to pick up as a monogenic trait or as a single gene. So here what I've shown is average age of onset for individuals harboring different genetic mutations. But what I'm showing in the next slide is that even within a specific gene, there's tremendous variability in when disease onset occurs. And I think what this tells us is that although we know that there is a strong genetic component in ALS, there are other modifiable factors that impact the way that disease presents in terms of the timing of onset, and we think also the rate of ALS progression. Several factors have been associated with modulating the onset and development of ALS historically. These include things like exposure to chemical toxins, body mass index and obesity, smoking, occupational hazards like exposure to radiation or high voltage electricity, as well as viral infections. And as someone whose real expertise is in the area of infectious disease and immunology, it's viral infections that I'm going to focus on today. The reason for that is because, sorry, some issues with my animations here. The reason for that is because there's been a really long history of viruses having been associated with ALS or ALS-like diseases. Everything from poliovirus to herpes simplex virus to human endogenous retroviruses, which are really viruses that form a natural part of our DNA and have integrated into the human genomes over the course of millennia. The issue, though, is that historically, neurologists and other experts who tended to focus on studying ALS haven't worked very closely with people in the infectious disease field. And that's, I think, led to a lot of confusion about what, if any, association there is between viruses and ALS and other neurodegenerative diseases. And one of the major issues that was immediately evident to someone who studies viruses for a living is that a lot of the viruses that had been historically associated with ALS have been associated because of an issue that we call detection bias. All of the viruses that I just mentioned cause chronic disease, essentially. These viruses, once they infect us, stay with us for our lives. But the number of viruses that do that are relatively rare. Most viruses, things like influenza or even SARS-CoV-2, make us sick, our immune system responds, it gets rid of the virus, and then the virus is gone. The problem is that it's very difficult to associate changes in ALS or any chronic neurodegenerative disease with an event that happened potentially in the distant past and which is now gone. So, you know, if an individual was infected with flu 5 years ago, 5 years later develops ALS, there would be no logical reason why you would necessarily associate that individual's ALS with an earlier influenza virus infection, because, you know, everybody gets infected with influenza. And so the question that we wanted to ask is, first of all, why has there been this historical association of viruses with ALS and other neurodegenerative diseases? Is there a common thread? Is there essentially a common reason or way that viruses might influence disease? And if we understand that, is there a way that we can develop more effective treatments or preventative measures to prevent disease progression? So we know now with increasing amounts of depth and clarity that common acute virus infections, so things like the flu, the common cold, they can impact our neurological system. And some of the ways that this happens is that viruses that cause infections in other tissues, like the lungs, for example, during viral infection, the response to the virus results in inflammation. And that inflammation, those inflammatory markers, can circulate through our blood and have impacts at distal sites in the body, including in the brain and in the spinal cord. It can result in activation of immune cells in our brain and spinal cord that are meant to protect us, but if they're given the wrong signals, can also harm our neurons. And it also can result in infiltration of immune cells into the brain and spinal cord, which we normally consider immune privileged sites. A great example of this has been something we've all come to know quite well in the popular press over the last couple of years, which is that approximately 33, and this is likely an underestimate, of COVID-19 patients experience some neurological symptoms that persist after their infection. Examples include things like loss of taste and smell. This was in the news a lot early in the pandemic. dizziness, but also things like stroke and encephalitis, which are much more rare. So infections that occur in sites outside of the brain and spinal cord can still impact the brain and spinal cord indirectly. And that's really, really important, we think, and is an issue that we really wanted to tackle. One of the reasons that we thought it was plausible that viruses might impact ALS is because a lot of the things that go wrong in cells in patients living with ALS are also things that viruses make go wrong in cells. As an example, we know that we can see mitochondrial dysfunction in many cases in various forms of ALS. We see chronic inflammation, including neuroinflammation. We see toxic protein aggregation and spread, things like TDP43 aggregates or SOD1 aggregates. All of these are also things that viral infections can do to cells. And so because there's this intersection whereby viruses perturb a lot of the same processes within our cells that we know are sometimes altered in ALS, we thought that it would be important to understand how these two things come together. And of course, there are lots of rare viruses out there that cause, really severe neurological outcomes, but we didn't feel like that was a particularly pressing question to look into. What we really wanted to know is if common everyday viruses that we all get infected with can impact ALS. And if so, what's the mechanism? And if we understand the mechanism, are there ways that we can intervene to either prevent or slow ALS progression? What I want to say at the outset so that it's clear is that we don't think that viral infections per se cause ALS. We think that what likely happens is that individuals who have genetic susceptibilities may have their ALS triggered or progress more rapidly after a viral infection. So we think this is a combination of factors whereby there has to be sort of an underlying genetic risk factor, which we may not know about. And in fact, in most cases, the current data in ALS would suggest that we don't know about. And that in combination with a virus, those sort of two hits may be enough to cause disease. So the way we went about testing this was through using a really common mouse model of ALS. And this is a SOD1 G93A mouse model. The reason that we did this is because these mice experience ALS-like clinical signs in a way that's very predictable. And we wanted a model that was very predictable so that we could understand how infection alters the course of disease in that model. So these animals start to experience hind limb weakness when they're about 3 months of age. And by the time they get to about 6 months of age, that hind limb weakness has progressed to full hind limb paralysis. So they experience sort of very typical motor neuron degenerative disease. The reason that this model is useful is that just like in patients suffering from ALS, before onset of clinical signs happen, these mice look completely healthy for all intents and purposes. And so the question we wanted to ask is, if we infect these mice with influenza virus during the stage of their lives in which they're healthy, would that later impact the qualities of ALS or ALS-like symptoms that these mice experience? So the way we went about doing this is we took wild type mice that do not develop ALS and these SOD1 mice that do spontaneously develop ALS-like symptoms, and we infected them with kind of a standard dose of flu. And then we did this test called a rotarod test, which measures the ability, essentially the functionality of the limbs of the animal. So what happens is the mice sit on what's basically a balance beam, and then that balance beam starts to spin. And healthy mice are really good at balancing on that balance beam. But for mice that are experiencing motor neuron degeneration in their hind limbs, they get increasingly less capable of balancing. And it provides a way for us to monitor how fast ALS-like disease is progressing in those animals. So the first thing we did was we just wanted to know, if you infect a mouse that has genetic factors that will lead it to develop ALS, does it respond any differently to that infection than a mouse that has no genetic risk of developing ALS? And what we found was that during the acute stage of infection, so when the mice actually get sick, they look just like normal, healthy mice with no genetic susceptibility. They lose a little bit of weight. That's very typical in mice that get infected with viruses like flu. So you can see this is the weight of uninfected mice. It stays really stable. Obviously, they continue to eat. They gain a little bit of weight. Mice that get infected, they lose a little bit of weight because they get lethargic. They get a little bit dehydrated, but then they recover. And the recovery of the SOD1 mice, which spontaneously develop ALS-like disease, and wild-type mice or healthy mice is exactly the same. We then went on to do even more careful assessments of what's happening with the virus in these animals. And so we looked at how much virus there was in the lungs of these genetically wild-type mice and these mice that develop ALS-like disease. And we found there's no difference in the amount of virus in their lungs. And when we took just cells from these animals to see if there's any difference in the way virus replicates in their cells, we also saw no difference. So viral infection in a healthy mouse versus a mouse that will go on to develop ALS are exactly the same. And this is in essence the heart of why this question has been so hard to study in humans, because someone who might later get ALS doesn't experience an acute infection any different from someone who will never develop ALS. Everything at the stage of the actual infection seems to look the same. But here's where things get interesting. When we start looking at how well the mice perform on this rotarod test, what we find is that the mice that go on to develop ALS, if they've been infected by flu, they perform much worse on this test much earlier than mice that develop ALS but have never ever been infected in the context of their lives. So the infection, many weeks after the infection has been cleared and the mice have recovered, starts to impact the rate at which their ALS-like symptoms progress. And if we look at how long it takes these mice to reach the point where they experience complete hind limb paralysis, what we see is that mice that have no genetic risk for ALS obviously never experience hind limb paralysis. In the solid red lines here, what we see is the timing for which mice that have the SOD1 G93A gene and spontaneously develop ALS-like symptoms experience full hind limb paralysis. And in the hatched red line, we see how long it takes the same mice who have been infected during the healthy stage of their lives to reach complete hind limb paralysis. And what you can see is that if the mice were infected during the healthy stage of their lives, they reach this point much, much sooner. So their ALS onset happens at the same time, but the disease progresses much more rapidly. So the obvious question was why? Now, viruses do two main things when they infect us. One is that they tend to kill the cells that they infect, and that can be a problem. But the other thing they do is they stimulate an immune response, right? And the immune response is supposed to protect us, and it does protect us in most cases. But sometimes genetic mutations and other environmental factors can cause our immune response to respond in a way that can also harm us. And so we wanted to know whether virus killing cells was important in the virus's ability to cause ALS to progress more rapidly, or whether it was the immune response that was primarily causing ALS to progress more rapidly. So the way that we tested this was we could take the influenza virus and kill it essentially, so that the virus can't actually infect the animal and cause cell death in the animal. However, the virus could still be recognized by the immune system. So this basically allowed us to ask, is it the virus, killing cells when it's infecting us that's causing ALS to progress more rapidly? Or is it the immune system's response to the virus that's going wrong and resulting in disease progressing more rapidly? So we injected mice with this inactivated fluid that can't replicate and can't kill cells. And what we found was that when we injected this in the hind limb, which is where disease actually occurs in these animals, it's limited to their back legs, these mice also progressed more rapidly than mice that have never seen virus before. However, if we gave the same dead virus into the mouse's lungs, nothing happened. Disease did not progress more rapidly. They looked just like animals that had never been infected before. So what this suggested to us is that it does indeed seem to be the immune response that is at least in part responsible for accelerating disease. But location really matters. If you induce inflammation in a major muscle, and of course ALS is a neuromuscular disease, that seems to be a problem. But if you induce only a little bit of inflammation that's just limited to another tissue or organ system like the lung, that doesn't seem to be a big problem. It doesn't seem to impact disease progression whatsoever. So we wanted to know next, well, what is going wrong with the immune response that's causing these mice who are infected during the healthy part of their lives to have their disease progress more rapidly? And in this mouse model, the area of the mouse where the ALS-like disease is most obvious is in the lumbar spine. And this is the part of the spine that's responsible for talking to the hind limbs of the animals. Unlike in human patients where ALS really manifests throughout the central nervous system, in this mouse model, it's really limited to the spine, and we still don't actually completely understand why that is. But in the spine, in addition to there being nerve cells, right, that help us sense things and help send signals to our muscles that allow us to do things like walk and move around, there are also all kinds of immune cells that hang out to try and keep these nerve cells healthy. But we know that in ALS, sometimes these immune cells can misbehave and lead to death of neurons. So we wanted to ask, well, does something strange go on with these immune cells that causes problems for neurons in the animals that have been infected? So what we did was we repeated the same experiment that I just talked to you about. The mice were infected at day 60, which is when they're healthy. And at day 120, which is sort of right before we start to see complete hind limb paralysis begin in this animal model, we looked at the immune cells in their lumbar spine and asked, is there any difference in animals that were infected versus animals that were never infected? And what we found when we did this is that microglia cells, which are one of the really important immune cells present in the brain and spinal cord, there was more of these cells in animals that had been infected with flu almost 2 months earlier, remember. So we're looking at day 120. The animals were infected at day 60 of age. So this is 2 months later, long after the virus is completely gone, that there still seems to be a problem whereby these immune cells are angry for all intents and purposes. They're activated. They think there's still an infection present. And we know that that can lead to death of motor neuron cells. We saw the same trend with astroglia, although the effect size was much smaller. And astroglia are sort of a related cell type that also, in general, sort of survey our brain and spinal cord and protect us from infection, and also sort of clean up death of neurons that die by natural processes. Now, because this animal's genetic susceptibility is related to a mutation in the SOD1 gene, we also wanted to look and see whether there was an increase in SOD1 aggregates, because SOD1 aggregation has been proposed to be one of the mechanisms that might contribute to ALS. There we saw no difference. So the earlier infection didn't seem to impact aggregation of SOD1, but did seem to have this profound and unusually long-lasting impact on the way that immune cells were behaving in the spinal cord. So what this tells us is that viral infections in general, the reason that they may have been associated with ALS in the past, and the reason that there seems to be association with so many different viruses that cause all sorts of different types of infections in different tissues, is because one of the things that all viruses have in common is the fact that they stimulate an immune response. In some ways, this is really good news, because if it were the case that every single virus impacted ALS in a vastly different way, it would be very difficult for us to develop interventions that might help prevent this effect from occurring. However, the way that viruses stimulate the immune system is largely conserved. And there are a lot of good drugs out there that we know we can use to help prevent this kind of inflammation from happening. Now, for the average person, we typically don't treat them with really aggressive drugs if they just have like a simple cold, right? I'm sure everyone in the audience has had the experience of, going to their family doctor, they've been feeling unwell, maybe they've had a fever or a cough, and the doctor says, it's a virus, antibiotics aren't going to do anything, go home, rest for 5 days or 7 days, and you'll feel better. So that's usually the case because a lot of the drugs that modify our immune system can also have side effect profiles that just aren't worth the risk for someone who's going to get over an infection fine anyway. But for someone for whom that infection might profoundly impact something like ALS down the road, using those drugs might be a really attractive option, actually, right? Because the issue is that people who have these genetic factors that lead them to be susceptible to ALS are having an intrinsically different immune response, which creates an intrinsically different risk than other people. In addition, in many cases, we do have antivirals that can be used to treat common viral infections. But again, we don't usually do it because for most people, those infections are what we call self-resolving. They don't resolve in severe illness. People have a cough or a runny nose for a few days and then they get better on their own. So we don't want to treat them necessarily with drugs that have, the possibility of side effects, even if those side effects are very rare or very mild if we don't have to. However, in a situation whereby treating that infection more aggressively might prevent the onset or more rapid progression of a disease like ALS, those kinds of differential clinical decisions could be made. So what our group is really interested in right now is looking to see which classes of those drugs are most effective in preventing this increased disease severity that we see in the animals that were infected. And then once we have a grasp on that, working towards clinical trials to test whether some of those interventions will also be successful in preventing these same kinds of things happening in humans. The other thing that we're really interested in understanding right now is whether or not the phenomenon we're observing applies to all types of genetic backgrounds that lead to ALS susceptibility, or whether this is something that's more limited in scope, for example, to to individuals who have mutations in SOD1 or maybe mutations in TDP43. That's going to be really important for us moving forward so that we know who to target with these potential interventions going forward. Oh, I'm sorry. I'm just going to pop my screen back up for one more minute. So with that, what I want to do now at the conclusion is just thank the people in my group who really did this work. The work I've talked to you about today has really been spearheaded by an extremely talented PhD student in my laboratory whose name is Art Marzak, as well as now Dr. Jonathan Mapletop, a former PhD student in my lab who has gone on to work for a pharmaceutical company right now. Braden Calborough, a former master's student, Andrew Chen, a current medical student, and Vithushan Surendran, an MD PhD student, are also involved in various aspects of this work. We've been really lucky to work with some incredibly talented collaborators and to receive funding from a variety of organizations to support this, including ALS Canada. So with that, I want to stop to ensure that there's lots of time for questions and discussions. I hope that you found the presentation both interesting and accessible. Thank you very much for your attention.
Hi, Dr. Miller. Thank you. I'm James. I'm one of the student ambassadors, and myself, along with Brian, will be asking some of the questions that were asked during the chat during your presentation. I found it very interesting, especially, like you said, given the COVID day and age. I think we're all a little bit more well-versed in viral terminology. So with that being said, first and foremost, are there any types of viruses specifically thought to cause ALS more than others, i.e. retroviruses, influenza viruses, et cetera?
Yeah, so I think that's a really important question that we don't have a good handle on. In the medical and scientific literature, there are certainly viruses for which the association has been stronger historically. But in many cases, that historical link, I think, is a function of this detection bias that I alluded to at the beginning of the presentation. So often the way these associations are made is that people will look in post-mortem tissues and will say, okay, we are finding that this virus, let's say a retrovirus or a herpes virus, is more prevalent in the neuronal tissue of an individual who's passed away as a result of ALS relative to some control cohort, right? It's individuals who don't have ALS. However, you can't really do that for most viruses that, cause an infection and then are gone, right? Because they're just not there anymore. And that has been, I think, the major factor that's limited the ability of the medical field collectively to really understand if and how viruses influence ALS. So our goal was really to use this model to understand if all of these associations have been made, but there was really no understanding of whether the associations were causative. And so that's a critically important distinction in medicine and science, right? The difference between association and causation. And so we think that these studies in animals provide at least a causal link in terms of a biologically plausible mechanism through which a variety of viruses can do this. I think it's likely, James, that certain viruses might do this, might be a higher risk than other viruses, but I think it's quite likely that many viruses are capable, and we just don't yet know to what extent. It will take some time to sort that out.
Hi, I'm Brian, the other student ambassador monitoring this meeting. So next question is, could you expand on the SOD1 mice to IAV infection? Were these SOD1 mice ALS positive, or do they just have SOD1 gene susceptibility?
Yeah, great.
Also, second question, how do you give a mouse or another animal ALS? Probably the same question, sorry.
Okay, so the way that these mouse models work in the ALS field is typically genetic instruments are used to introduce a known mutation associated with ALS in humans into the mouse. In the specific case of the SOD1 G93A mouse, the SOD1 G93A mutation is a mutation in SOD1 that's known to lead to ALS in humans. And so what's happened is that in this mouse model, these mice express this ALS-associated version of SOD1, and all of the mice who have that gene spontaneously develop ALS starting at about 3 months of age. And that's why that model is useful for us as scientists who study ALS, because we know that normally these mice start to experience disease around 3 months of age, and disease sort of progresses to complete paralysis by 6 months. That reliability allows us to test in a way that we can monitor very closely if something either slows disease down or speeds disease up relative to what the mice normally experience. And so in our study, what we found is that the mice that were infected prior to their ALS onset happening ended up having much more rapid progression of their disease than the exact same mice who still develop ALS-like symptoms spontaneously, but have never been infected before.
Very interesting. To go specifically back to your presentation, maybe do a little bit of a biology refresh. What exactly are the roles of microglia, and have the levels of microglia and astroglia been measured in humans with ALS versus humans without? And if so, what were the results?
Yeah, all great questions. So obviously ALS itself is very heterogeneous, as I discussed in the talk, right? And we have certain subtypes of ALS that are more frontotemporal dementia-like, and on the other end of the spectrum, much more classical, purely neuromuscular ALS, and then everything in between. In humans, we do know that this chronic activation of microglia and astroglia is a hallmark of not only ALS, but lots of other neurodegenerative diseases that are, closely related to ALS, things like Alzheimer's disease, Parkinson's disease. And what's a little bit unclear is why that inflammation is happening, whether the inflammation itself is causing motor neuron death, or whether the inflammation is a response to something else that's going on that's contributing to ALS. So in the CNS, microglia and astrocytes, the role that they play is really, they're like guards. They're the guards of the immune system. They perform what's called immunological surveillance. So they're looking out for things like viruses or bacteria that, if they got in our brains or spinal cords, would make us really sick. So normally microglia are friends. They do really important work cleaning up damaged cells and eliminating infections. What seems to be happening in the model of ALS we're looking at, though, is that the infection is making the microglia misbehave in a way that they think the infection's there even when it's not there. And the things that microglia do to protect us from infection, if those processes last too long, they can actually harm our own cells. We see that not only in the context of ALS, but in the context of lots of conditions. Actually, in COVID, most people who become severely ill from COVID don't become severely ill as a result of the virus physically damaging, let's say, their lungs, for example, or other organs in their body. Their immune system responds too strongly. And that's why people who become severely ill with COVID, we tend to see that happen like quite a while after they initially experience symptoms. It's often sort of like 2 weeks after they've been infected. And the reason for that is because the immune response to an infection peaks at about 2 weeks. And so that's where we see, people ending up in hospital on respirators, for example. It's because of this concept known as immune dysregulation, where our body's immune system, which is normally designed to protect us, can sometimes hurt us if it's not regulated properly.
Very interesting. Thank you for insight. So, with your research, have you in your lab studied neutrophils in ALS patients?
Yeah, great question. So we're really interested in neutrophils generally because they're the most abundant type of white blood cell or immune cell in our blood. So there's a lot of neutrophils. And neutrophils are also really important because they can create a lot of this inflammation. So we haven't specifically looked at the contribution of neutrophils to inflammation or ALS onset in our model, but it's a really interesting question that we're very interested in exploring in more detail. So one of the big sort of next steps for us is to look at the specific immune populations that may be talking to the microglia and making them misbehave. Because again, there are lots of available drugs already approved on the market that can target some of those specific populations and may help us to intervene in a way that's helpful.
More research needs to be done,
Yeah.
as always. Specifically, is there a connection that maybe you've seen or read up on with EBV/cat scratch disease, Bartonella infections?
Yeah, so I mean, in addition to viral, we studied viral infections because my group's expertise is really strongly in virology, but I think it's very likely that what we're seeing is not limited to viruses. So there are lots of other types of infections, and especially those caused by various types of bacteria that may also contribute to ALS. And in fact, other groups, both here in Canada, but also internationally, have addressed this experimentally in the past too. So bacterial cells are made of components that look different from our own cells. And those components can be recognized by our immune system. It's how our immune system knows if we're infected, because it's really good at distinguishing what cells in our body look like from bacterial cells or viruses. If you take just a component of bacterial cells called LPS, it's called lipopolysaccharide, and it's like a soap-like molecule, and you inject that into these SOD1 mice, it will also speed up the progression of ALS. So inflammation more generally seems to be a concerted mechanism through which ALS progression is sped up in the context of infection. And again, you know, I try and take really a positive view on this as it relates to thinking about implications, because, you know, there are more viruses and bacteria out there that are capable of infecting humans than you could almost count. And if every one of those behaved differently, our ability to target all of those vast infections individually would probably, at minimum, would take a very long time. At worst, it just simply wouldn't be feasible. But because they all impact the immune system in a fundamentally similar way, that creates a real opportunity to intervene therapeutically in a way where we don't care exactly what the infection is caused by, but we might be able to treat in the same way and still have the same outcome.
That makes a lot of sense. Different pieces moving, but the same commonality.
Right.
Perfect. Next question is, what are your thoughts on inhalers that prevent the release of substances in the body that could cause inflammation?
So inhalers actually look good to us, right? So I think one of the really interesting experiments we did that I talked about in my presentation was an experiment where we killed the virus, so it was no longer able to actually infect our cells, but it still elicited an immune response, right? And the point of that experiment was to determine whether it was the virus or the immune response the virus was causing that was leading to disease happening more rapidly, right? Or progressing more rapidly. And what we found is that the inflammation in the muscle, right, which is impacted in ALS, right? It's the ability of our motor neurons to talk to our muscles ultimately that's impaired in ALS as a result of motor neuron damage and death. That's a real problem. But when we elicited the same inflammation, but it was localized outside of the muscle, right, just in the lung, we didn't see the same progression of disease. Now, there are important differences between this mouse model and humans that experience ALS, right? And that's critical to remember, because in this mouse model, as I told you, the disease is really limited to the hind limbs and the lumbar spine. In humans, obviously, we have both limb and bulbar onset ALS, and there is profound respiratory involvement, right, for many people, especially those who experience bulbar ALS. But even people who experience limb onset ALS, right, usually end up having respiratory involvement at later stages of disease. What it tells us, though, is that location really matters. And I think anti-inflammatories, right, are actually what's most often delivered by inhalers currently. Things like steroids that are designed to combat inflammation. And the idea would be that those might be very positive. What I should say is that some of you in the audience might know that there actually is a long history of clinical trials testing anti-inflammatories in ALS. And they have been very disappointing in terms of the results. None of them have shown any major efficacy. But I think the reason for that is because of the way the trials were designed. In most cases, the trials were designed to give anti-inflammatories to people whose ALS had already onset, right? We were trying to treat people who had ALS with these anti-inflammatories. And the reality is that for some of these anti-inflammatories, that may be too late. What we might need to do is better identify people who are at risk of developing ALS. Right now, that's more straightforward for familial ALS, but we're getting better and better at doing this even for sporadic ALS, because very soon, I suspect we'll get to a point where when people are born, their whole genome will be sequenced as a matter of routine, in the same way that we do sort of typical health checks on neonates or babies when they're born for a variety of isolated diseases. Now it's becoming more and more feasible to do whole genome sequencing. And in cases where some known genetic risk pops up, that may allow us to intervene, as I said, differentially while those people are still healthy, knowing that that might ultimately impact whether or not they develop ALS, because in some people, they may have a gene that by itself is a risk factor for ALS, but does not guarantee an individual will eventually experience ALS. But some environmental factor, perhaps an infection, combined with that genetic predisposition might trigger disease. That's sort of the panacea, right? That's the stage we want to get to, where we can take a much more preventative approach, because I think what history has told us is that treating disease once onset has occurred is very, very difficult. I think there's a lot of hope out there in the clinical space about effective ways that we can treat disease in individuals who are already experiencing ALS. But ultimately, what we want to do is get to a point where we can prevent individuals from having onset happen to begin with, right? And so both things need to happen in concert. But I think obviously prevention is better than treatment, because treatment means that the person has to have ALS to be treated. What we'd like to do is prevent it insofar as possible before it even starts.
It's amazing. You start to answer questions that are coming up on our sheet. So I'm going to expand on one of the ones that was asked. You mentioned that, when you tested these animals, it was the limb onset version. How do you test bulbar onset ALS on animals?
Yeah, there's not a great way right now, because there's not a good animal model of bulbar onset. We can do other things. So there are some other animal models of ALS where ALS starts in the brain more so than the spinal cord, and that more resembles bulbar onset. The problem is that most of those animals don't ultimately experience motor neuron disease. Nevertheless, there are other things we can do to sort of look at other features of ALS in those animals. Some of those animals actually experience symptoms or clinical signs that are more consistent with frontotemporal dementia. So that's one thing we can do. We can do more sort of cognitive tests. Ultimately, though, I think what we really want to do is, as a next step, ensure that what we're seeing in animals is actually true in humans. And that's a very non-trivial task. It involves following people for a long time. But it's really, I think, the next important step in understanding the contribution of infections in ALS more broadly.
Thank you. So next question is, have there been any studies conducted on potential antibodies for certain viruses among those in ALS reversal cases?
Yeah, great question. So there haven't been any good studies of the impact of antibodies in the context of reversal that I'm aware of. What would be really interesting to know is, and something we're studying, in fact, is let's say you're immunized against flu, right? But have a genetic predisposition towards ALS. Can things like immunization, which can prevent infection from happening, lower your risk, right? Because that's a fantastic way to intervene potentially very safely, right? There are also certain antibody drugs out there, like monoclonal antibodies, that have been discussed a lot in the context of the current COVID pandemic, that could be used to treat infections in individuals who are high risk. As an example, potentially people who are at risk of developing ALS. And so understanding those things are really important. The other thing that will be important, I think, is understanding if there are individuals who have ALS for whom infection doesn't change the course of their disease, because those people may have intrinsically different immune responses that we could learn something from in terms of, what is a healthy immune response in an ALS patient versus what is an unhealthy response. And then again, design, new drugs or new interventions to try and ensure that people have these healthy, beneficial immune responses and not the adverse consequences of immune responses that are dysregulated.
I do want to be respectful of your time, so I believe these next two questions will be the last ones. We've just been flooded with so many. It's been a phenomenal presentation. The second to last question is going to be, is your research specific to genetic ALS only, or have you explored sporadic ALS with viruses as well?
Oh yeah, good question. So right now, again, the difference between familial and sporadic ALS from a genetic standpoint is really minute. In fact, in most cases, there probably isn't a difference. There are some genes that have only been associated with sporadic ALS, but it's likely that if we continue to look hard enough, we'll also find that those genes contribute to familial ALS as well. I think what we're really interested in doing is determining whether there are situations whereby an individual has a gene which alone would not lead to ALS, for which a viral infection or some other environmental insult might trigger ALS. And so in the mouse models that exist currently, it's typically the case that sort of like all of the mice that have these ALS associated genes end up developing an ALS-like disease or have a conserved set of symptoms and clinical signs. We don't yet have what we would call an inducible model of ALS. And that is a critically important area of study because, again, in the vast majority of cases, we don't know what the genetic basis of ALS is. And one of the reasons for that might be that it's not only genetic, right? In fact, I think it's very likely that in most cases, it's a gene plus something else that could lead to disease onset. And so we have some leads that we're looking into to study that, but it's still too preliminary to get people too excited about yet, because we're just not certain, but it's something that we're working really hard to understand.
We want to thank you for your time. And as James mentioned, this year, the last question, respect for your time. So where do you think the research on viruses and ALS needs to go next?
Yeah, great question. So I think the first, there are a couple of things that need to happen in parallel. One is that one of the things our group is working to do is, again, understanding whether or not what we see applies broadly to all sort of all ALS, regardless of what the genetic mutation or predisposition is, or whether it's limited only to certain types of ALS with very specific mutations, like SOD1 associated ALS or TDP43 associated ALS. So that's really important. And then even more importantly, at the end of the day, right, we're still working in mice, and they're great as a model because, you know, we can do really careful experiments to assess things like causality, right, that we could never do in humans. But we need to do the really hard work to really understand if what we're seeing in mice is also going on in humans. And once we know that, then I would say the priority would be to look at these candidate drugs that are already on the market and approved to treat inflammation in various ways, and see whether or not those can improve disease for individuals who have it already, and potentially prevent disease in, for example, cases of familial ALS, where there's a really well-known genetic risk factor. If I may, I'm sort of half monitoring the chat, and there's a really important, I think, discussion happening that I'd like to address, because it does speak to, I think, what's a really important concept in the context of the presentation. So Matt asked, in our model systems, we used a killed virus, and in certain cases, vaccines can be composed of killed virus. And so the question is whether or not vaccines might be problematic or not in certain cases. And I think it's a really important question. The way that we prepare the dead virus for this study is, first of all, much, much different than the way vaccines are prepared. Right now, in terms of like sort of how clean they are, right? When we're doing this in animals, the idea is just to kill the virus. When vaccines are actually made, they're produced right in a much more carefully controlled way that results in a product that's a lot different than what we make in the lab. Nevertheless, it is important, I think, for us to understand if certain types of inflammatory triggers, including vaccines, are helpful or harmful. Right now, I think the vast majority of evidence would suggest that they're helpful, because vaccines, when you give a vaccine, they typically generate like sort of this much inflammation. If you get infected by the same pathogen, they trigger like, you know, this much inflammation. So even if there is some small risk associated with vaccines, I think the chances are that by preventing the actual infection, the net benefit would be positive. Nevertheless, it may also be the case that certain types of vaccines are more appropriate for ALS patients than others. For example, in our studies, we gave what amounted to a vaccine right up the nose versus one that was injected into the muscle. Well, for influenza, there is a nasal spray version of the flu vaccine, right? In addition to the typical flu shot. So understanding whether for individuals who have ALS or who are at risk of developing ALS, there might be certain vaccines that are better than others. That's also an important question, but it requires much more study. So at this point, I think all of the data would suggest that infections are much more harmful. And so until we have a better understanding of that interplay, the current evidence would suggest that there is a net benefit to preventing the infection. And so I would still encourage people to get infected until there's, strong evidence in humans that would suggest otherwise.
I'm glad you touched upon that because that was starting to come up quite a bit in the chat. So I do appreciate you taking the time for that one. I do want to be respectful of your time. I want to thank Dr. Miller for giving a fantastic presentation on viruses, inflammation, and ALS. This is going to be the portion of our forum that's non-recorded for everyone to speak freely. Dr. Miller, you're more than welcome to join, but we understand you've got a lot going on to benefit the world. So if you need to go, that's all right. And I'm going to turn this now over to Mick Finn.
Yeah, thanks very much to both of the student moderators, and again to all the organizers and staff at Everything ALS for the chance to speak to you all today. Really appreciate it. Again, I hope that you enjoyed the presentation and found it understandable and accessible. That can sometimes be a challenge for us when we're used to talking to colleagues who speak medical jargon, but I hope I was able to present it in a way that everybody found was easy to digest and understand. It's always, a real pleasure to be able to speak to organizations like this, where we have an opportunity to interact with individuals and their families who are experiencing ALS. And I think it's really critically important. And so thank you all so much. And I wish you all the very best.
Thank you, Dr. Miller.
Thank you, Dr. Miller.